And I'm Dalaena Rivera. I'm a second-year PhD student in the Dr. Robert Llewellyn's lab at SDSU. And today I'm going to be talking to you about the bacterial adherence in the intestinal epithelium of C. elegans. So to bring it out broadly, so we know that the human gut microbiome consists of good bacteria, fungi, and viruses. And when an invading pathogen comes in and upsets the balance of the microbiome, it can cause a big disruption. And this disruption can lead to many gastrointestinal diseases such as obesity, type 2 diabetes, and even depression. And one way that the host, us humans, can help protect against these invading pathogens is through negative selection pressures, in which the host immune system can help fight off these pathogens. And the less understood positive selection pressures are when the host can cause the colonization of these good bacteria over the pathogens. And however, studying the human gut microbiome is really difficult sometimes because there are so many complex and diverse interactions that are happening between all of these different microorganisms and the host. And so C. elegans, which is a tiny microscopic nematode, has emerged as a powerful tool in which we can study the microbiome. And that's because the intestines of C. elegans are structurally and functionally similar to humans. And my lab will often go sampling out into the wild to look for wild C. elegans strains. And then we do that by going out into nature. We look for rotting stems, rotting fruits, take them back into lab. We put those onto plates that are seeded with E. coli. And this E. coli acts as a bacterial food source for the worms. And so over time, the worms will crawl out from the fruit or those rotting stems and feast on that bacterial food source. And then from there, we can pick these worms off and do a whole bunch of different experiments with them. And from sampling, we actually have found some novel bacteria that seem to be adhering to the intestinal epithelium of the worm. And so if that's the worm, these bacteria will directionally bind to the epithelium of the worm, and they'll bind to the entire length. And they bind really, really close, and they kind of look almost like bristles, like on a toothbrush. And so one way that we can study these worms for my project is looking at this interference. If this is the intestinal epithelium and this is the bacteria, what is happening here that mediates that adherence phenotype? And one way we can test that is by taking some worms and mutating them through a chemical mutagen, and then colonizing the worms with whatever adhering bacteria we want. And hopefully, one of the mutants will not be colonized. And that indicates that whatever mutation that worm received is most likely responsible or involved in the bacterial adherence. So then we could take this worm and do sequencing to figure out what that gene is. And the big picture of my project is looking at these worms one important thing to note is that one of these worms, when colonized with one of the adhering bacteria, is pathogenic, or will exhibit pathogenic effects. So this worm will be sick when colonized with this pathogen. And these other two worms will behave as normal. since that adhering bacteria provides a neutral or commensal effect. And we're trying to see if we can possibly promote the colonization of these commensal bacteria as opposed to these pathogenic bacteria to protect the worm. And by doing that, we can possibly understand mechanisms in which humans can protect themselves by promoting the colonization of good or commensal bacteria over pathogenic bacteria.